that, our next speaker, for me it's an honor to introduce Dr. Rob Knight with the talk from University of California at San Diego, with the talk Microbiota Analysis from 36,000 feet high. Please, Rob, go ahead.
All right. Thanks. I'll just start the screen sharing. First off, I'd like to thank Gloria and the rest of the organizers for organizing this wonderful meeting and for inviting me to it. It's GloMiNe, I think, is really providing a really important resource for the community. So Gloria had asked me to give an overview of microbiome analysis using our tools, and the backstory to this is that microbiome science really exemplifies what used to be a data-limited science, moving to a science that's instead limited by analysis. And my labs contributed substantially to this over the years. Back in 2008, we introduced a protocol that allowed us to simultaneously get insight into hundreds of microbial communities from different environments, including the lung, the rivers across North America, the air, an incredibly low biomass environment, and the Guru Negro microbial mat, still the most complex microbiome we know of. And through the technique that's now standard of barcoding these samples with individual error-correcting barcodes at the library prep step here in PCR, we were able to get a simultaneous analysis of hundreds of samples. But what was remarkable about this is that we doubled the number of 16S sequences in GenBank in this just by doing this one study. And the 500,000 sequences we collected, at the time, the Sanger facility down the hall from my lab at Boulder was still charging $8 per read. So it would have cost $4 million to do it with the old technology. With the then new 454 technology, it cost $12,000, and we did it overnight. And this is what opened up large-scale projects like the Earth Microbiome Project, the Human Microbiome Project, and so on. But what's most amazing is that amount of sequencing that would have cost $4 million in 2007, $12,000 in 2008, today costs us just 75 cents. And so what's really important at the moment is not so much decreasing the cost of sequencing, but decreasing the cost of the rest of the things in the workflow, so that so that all of the other so all of the other steps can be just as cost effective. And I'm not going to actually not going to say too much about that, but one thing we're right in the process of doing is moving field sampling from this sort of format of tubes to this format, where each tube is pre-barcoded, and the whole rack can be scanned at once. So the idea is that the future of this will be scanning whole racks of these tubes at a time, and this is something we're validating right at the moment, which will achieve similar cost decreases in other parts of the chain. So the great thing about this is that you can collect unprecedented amounts of sequence data concerning the microbiome, but then the problem with it is that you have to analyze all of that data. And here I'm showing you just a snippet of the first file from the Human Microbiome Project. And despite the fact that what we're fundamentally doing is ecology with this, it's really hard to ask ecological questions of who lives where in the environment and why, just by looking at the sequences. And this is increasingly a problem, not just for research, but clinically, because it's certainly your nightmare as a physician that someone's going to come up to you and say, I have this great news, which is I had my microbiome sequenced, and now I have this list of 1,000 species that they found in my gut, or maybe with shotgun metagenomics, 1 million genes that they found in my gut. And with all this data, you can tell me what's wrong with me, right? And I mean, what are you going to do? Refer them to your colleagues in psychiatry for being crazy enough to think you could do something with that in the 8 minutes you have together? So a lot of the challenge is to take all that sequence data and turn it into something that's useful. So a lot of what we've done over the last 15 years in my lab is essentially making that pipeline easier, to make it not crazy that you could take a new sample, get the sequences from it, and integrate it with other samples. And so this workflow is now standard of making barcoded libraries, mixing them together and sequencing them, demultiplexing them, trimming the barcodes, grouping the related sequences, building trees, and then using clustering techniques on those trees. And so we've swapped out successive pieces of the technology over the years, but the Chime pipeline essentially allows you to do all of this, both for 16S and for shotgun data now. And at UCSD, this is just one of a large number of different bioinformatics tools that members of the Center for Microbiome Innovation, which I direct, have contributed to. So we're really doing a lot on algorithm development. So in the time I have, I can't really explain how to use all this stuff. You know, applying all of these tools is very complex. It's like being in the cockpit of a plane and flying it. And so I can't tell you how to do that in 15 minutes. But what I can do is give you the safety briefing. So essentially telling you what you can apply and what you should watch out for. So one really important principle is listening to crew member instructions, as it were. And each microbiome tool is designed for a specific purpose. And it's really important to read the documentation and the papers by the authors of those tools and listen to your colleagues who use them frequently, because each of these tools is going to have a very restricted application area where it works well. For example, for Chime2, there's very extensive user documentation and starting to be developer documentation as well. And the three papers here are still the best resource for best practices for analyzing microbiomes. The first one's targeted at clinicians. The second one is more general. The third one is more targeted at mouse work, while they were in the process of updating these at the moment. Second important principle is to have a clear exit plan. And what you really want to do is you want to structure your data so that it's going to be clean with an easily testable hypothesis. So, for example, this picture I very frequently show of C. diff patients, where their stool samples are totally different from the stool samples of anyone who's healthy in the HMP. And if I animate them after fecal transplant, you can see that they go from the unhealthy region of the plot where those sort of spiky symbols are into the healthy region very clearly and immediately. That's the sort of structure that you want to be able to see, and you're only going to get that if you know what other data sets to combine it with, and you're asking a very clear question. So if you have a clear hypothesis up front, like in this case, that people are going to start off in a sick state, they'll move to a healthy state after the transplant. Structuring the analysis to test that clear hypothesis is going to make your job a lot easier, both for statistical tests and for visualization. Being prepared and alert will lead to better outcomes. And for example, being able to do different classifications can be really helpful in structuring your data. But there's a lot of choices at each of these steps. And so Chime2, Cheetah, Jupyter Notebook, and other emerging technologies automatically track provenance and let you reproduce the workflow and understand what happened afterwards. So sort of like a black box for your data analysis, and also allow you to reproduce it easily on new data that you feed in. It's really important that you don't ignore issues with your data. Like, you don't want to just stick your head in the sand and pretend those problems don't exist. And one very frequent problem is batch effects across studies. So, for example, in TCGA, the Cancer Genome Atlas, Greg Poor, one of my MD PhD students, found that if you just do the analysis, just do the analysis of the microbes as they appear in the samples, you see a very strong effect according to which site produced the different sequences. Whereas if you detrend for that, for that sequencing center effect, then you can start to get nice ROC and precision recall curves that give you a good classifier for one cancer type versus other cancer types. And so being able to look at those problems with exploratory data analysis and figure out how to detrend them is a really important initial step, because if you didn't know that problem was there, the analysis would be entirely driven by the sequencing center and not anything about the biological system. One thing I should note here is that very frequently differences in lab and computational methods between studies are much larger than the biological differences that you want to find. And so you have to be especially careful when you're combining different studies from the literature. Although Jamie Morton, one of my former PhD students, did a lovely paper in Nature Neuroscience last year rather, showing how you could do that in the case of autism, using a reference frame-based approach. And then finally, and perhaps most importantly, if your data are on fire, don't continue. And what I mean by that is a lot of data sets look like this, where there's just no where there's just no apparent clustering. So here we're looking at a lack of differences between males and females in a particular plot. And there's a lot of data sets that are like this, where they contain no pattern. And I've seen a lot of collaborators put grad student after grad student or postdoc after postdoc on a data set for years and years, trying to see a signal that's not there. So it's always important to be willing to give up on a particular hypothesis as just not being true, and look at your data in other ways, or go off and do a different experiment. So with that, I wish you a good, safe microbiome voyage as you're doing your own explorations. I'd like to thank a lot of people currently and formerly in the lab, huge number of collaborators across the Earth Microbiome Project and others, and the tens of thousands of members of the public who've contributed to our citizen science efforts. And thanks. Be delighted to take a question or two if there's time.
Thank you very much, Rob, for your interesting talk.